So I'll be talking about the touch DNA evidence in the Massereene attack. And this is the abandoned, burned-out car that we heard about from Justin, from which the different sorts of forensic evidence were collected. So this is what the evidence comprised. The DNA evidence consisted of swabs that were taken from a passenger side belt buckle inside the car, mobile phones that you heard about that were in the center console, as well as matchsticks that were found in the car, but the DNA evidence that I'll be talking about was a matchstick found on the side of the road. So before I go into what happened, I'm going to take just a minute and explain some terms so you know what a genotype or a genetic type is. Otherwise, much of what happens next will be less clear. So in biology, as you know, we all inherit chromosomes. We have 2 of them. We have 23 pairs of chromosomes. Each part of the pair comes from one parent, right? Most of us have 2 parents. It was different for Adam, I guess, but most of us too. And then if we drill down on a chromosome and take a look at what is in the chromosome, a chromosome it's sort of like a tape or a disk drive, and you unravel it, and has information in it. Instead of having 1s and 0s, it has a 4-letter alphabet of DNA, A, C, G, and T, that encodes essentially as a 3 billion letter instruction manual for how life operates, how you build cells, maintain them, repair them, and so on. So if you take a look at one location, which is called a locus or locus, then since we have 2 parents, we have 2 copies, and there are 2 different sentences that are sitting there, and those sentences are called alleles or alleles. Okay, so here are the 2 sentences, and the alleles that we care about in forensic DNA are taken from junk DNA, and they're just repeating units of 4 letters, ACGT, ACGT, some number of times. If that pattern repeats 8 times, then there's a certain length that repeat pattern has, and the allele is 8. And then from the other parent, you may get the same or a different number, and maybe that's 9. So there's a length over here. And so the locus has 2 DNA sentences. The allele is the number of repeated words, 8 in this case, ACGT repeated 8 times. And a genotype, what our genetic type is for this sort of junk DNA, is those 2 numbers, say 8 and 9. It's the 2 lengths at that one locus. For genetic identification systems, people, forensic scientists will look at 10 or 15 different loci that are contained on typically different chromosomes, so they can be independent. And that way we get more information. And the idea of this is that if you have maybe 10 or 20 different lengths that are possible at one locus, then there may be 100 possible pairings, 8, 9, 8, 10, 7, 13, and so on. So there are a lot of possible values. And that gives a distinguishing feature at that locus. And if you have 10 of these distinguishing features, then each person's genotype is relatively unique compared to everybody else in the population. And that's the basis of DNA identification. Okay? Make sense? Okay. So that was the main biology lesson. If you have that, we're almost there. But there are 2 extra ideas that are happening here, because this is touch DNA with DNA mixtures. So I have to tell you something about touch DNA. So let me tell you about touch DNA. If you have a reference sample with a lot of DNA, say typically 300-400 DNA cells, then if black represents one allele and white represents the other allele, then there's a lot of alleles sitting there, right? There are hundreds of them. So if you want, in the laboratory, what the laboratory does is it samples the DNA. It does it in several stages, you know, with the fluid and with the amplification and on the detection. It's sampling from the amount of DNA. So if you reach your hand into that bag think of the alleles as marbles, right? And you pull out some number of them, and there are a lot of them, then you're pretty much going to get a balanced number. The number of blacks you're going to get, the black alleles, will be about the same as the number of white alleles because you're sampling from something that's large. That's normal DNA. That's not touch DNA. Touch DNA is when you have very few alleles. Maybe you have 3, 4, 5 cells that are present. And now when you reach in and you just take a small sample, if you reached into a can of marbles, you might get 3 black ones, 1 white one. You can get an imbalance. There's a lot more uncertainty because the number is smaller. And so what you end up seeing in the data is the uncertainties reflected by peak heights or quantities, they'll turn into peaks in a minute, where the quantities can be a lot more of one allele than the other. It's not balanced anymore. In fact, one of the alleles can disappear completely. They could both disappear completely because there's a small amount and you're sampling from a small amount. Okay? So that's the concept of touch DNA. It's a small amount, and because the sampling that happens in the laboratory, you get less certain data, and we enter a realm of probability now. Things aren't as certain as they were before. So how does a laboratory overcome this certainty with touch DNA? Well, what the Cellmark lab in Abington, UK, it's near Oxford, does, let's see what they did with the seat buckle. On the seat buckle, they took the swabbing of the DNA and they sampled it 3 times, A, B, and C. They did 3 different tests on them. Why would you sample it 3 times? Because if in nature you have an undersampling, right, with more variation, like we see over here with uncertainty, then in the laboratory what science can do is to take more samples to try to regain some of that certainty. So instead of sampling it only once, which is what you would do with normal DNA, we sampled 3 times. And we'll test 1, test 2, test 3. I'll refer to those as A, B, and C throughout the talk. Now, with the cell phone, there was even less DNA. So in addition to sampling the touch DNA, that was a combination of DNA from all different parts inside the cell phone, in addition to sampling it 3 times, A, B, and C, that wasn't enough. So they did 3 tests and saw the signals were almost non-existent. So then they enhanced it. And what those next 3 tests, A prime, B prime, and C prime, do with enhancement is the laboratory concentrates the DNA to get it into more of it per unit volume. There's just more of it in there. And then they looked at it again and looked at the signals, and it still wasn't enough. So they did a second round enhancement. And what that second round of enhancement does is it then gets rid of more contaminants to make it even cleaner. And at that point, there was enough signal. So on this one set of swabs, 9 different DNA tests were done. On the matchstick found on the roadside, 3 tests were done, and looking at the DNA signals, which you'll see in a second, from those 3 samples, there was enough information so they didn't need to do any enhancements of the type they had done with the cell phone. Okay, so that's what's happened in the laboratory. Now, once you have data, this is what you have to interpret it. So what is the interpretation of DNA? I've already told you about touch DNA and how it's undersampled. Now I'll tell you about DNA mixtures. Actually, I'm just curious. Show of hands, who knows what DNA mixtures are? See, so they're not prevalent on CSI yet. All right. Most DNA evidence in many labs is DNA mixtures from sexual assaults, homicides, touch, doorknobs, and so on. It's not the case that DNA is taken from a crime scene off a doorknob or from a sexual assault. It goes into a laboratory, and before the next commercial break, there is a perfect profile matched against a perfect profile. Computers do something magical with it, and something happens before the episode's over. That usually, actually, that never happens. What real DNA evidence looks like, most of the time, is that you have a mixture of 2 people, 3 people, 4 people. If you have a hat that's shared by bank robbers, you may see 4 or 5 different people in there. Sexual assaults almost always have at least 2 people, sometimes 3. And so when you have a mixture, instead of only having 2 alleles, right, the black and the white, one from each parent, you start getting data that indicates there's more than 2 alleles in there. Well, one person's genotype can have at most 2 alleles. So if there are 3, there are going to be at least 2 people in there. It could be 3, and there's some number of people. So how is this interpreted? Well, suppose those lengths of molecules were 10, 11, and 12. Then, by looking at the data, the heights of these peaks, the patterns, and so on at this locus, as well as the other 9 loci from the different locations, what a scientist does, whether they're a computer or a person, the process of inferring information from the DNA data is identical. One writes out a list of these pairs of alleles. Now, in fairness to some of the lab people who are here, they may use a different procedure, but mathematically it's equivalent to what I'm describing. So they write out a list. Maybe 10, 12 is likely, 11, 12, 12, 12, and then they associate different certainties. Right? When there's uncertainty, we need to represent our uncertainty, and that's done using probability in science, which is probability essentially means how much we believe in each of those allele pairs, those genotype values, and those beliefs add up to 100%. That's what a probability is. A bunch of numbers that express your belief that add up to 1 or 100%. And at that point, no one has ever seen a suspect. A person hasn't, a computer hasn't. It's totally objective. We've gone from DNA to signals in a laboratory. This is a mixture because there's more than one person in there. To a genotype which has uncertainty that reflects the underlying ambiguity of the DNA data, and probabilities attached. And now finally, at some point later on, a comparison can be made between the evidence genotype, that list of allele pairs, and some known genotype, and a match statistic can be computed. And when you hear in the end, there's some number like a quintillion or a quadrillion, that's the result of this comparison, which we'll see a little more of. Okay? So that's inference. That's what, this is a very fast group. People can spend years learning this, and you haven't objected yet, so I just assume you're all fine. We'll move on. Okay. So this is what DNA data actually looks like. This is one of the 10 loci. And so this is what quantitative DNA data look like. They have different sizes that reflect the different alleles. So here we see a size that's a 9, a 7, 9.3. And each of these peaks corresponds to one of the alleles. The location of the peak is which allele it is, and the height of the peak reflects how much of that DNA was present from that one allele. So this would indicate that there's more at 7 than there might be at 9.3. It's called 9.3 because it's almost 10. And that's the way these things are designated. So that's what quantitative data looks like. At this one locus, they have names, THO1. And this is what the Cellmark data looked like for the matchstick at all 10 loci. In addition to THO1, we also see the other 9 loci here. They have names like D2, FGA. And this is the starting point for interpretation. One of the first things that we see is that the numbers on the side indicate that a height of 50 would be around here, and all these peaks are under 50. By usual manual review methods that people would do here in the States and in regular laboratories, they wouldn't even consider those data points because they're all under some fixed number, like 100 or 150. These numbers are all under 50. And unless you're doing some special analysis where you're doing more than one amplification, you wouldn't even look at that. So here's some issues with the data. The data have some ambiguity. First, we see mixtures. Here at this one locus, remember it was subdivided into several groups, 1, 2, 3, 4. And the first group, D3, there are 3 peaks here. And at D8, there are 4 peaks. So we have a mixture. There's more than one person in there. We also see over here that there's a very low DNA quantity. These 3 peaks at THO1, that's a blow-up of the same 3 peaks. it's a very low quantity. Those peaks aren't going over maybe 25, very small amounts of DNA, maybe only a few copies of DNA. Here, no DNA is visible, and at this locus, D21, we're seeing that there are peaks that are expected to be there for some reason. Someone might hypothesize, but they would have dropped out. So these are all well-known phenomena in the laboratory of dropout. It doesn't amplify, it's very low, or it's a mixture. Fortunately, the laboratory amplified the DNA 3 times, first, second, and third amplification, and they could compare, and they use a consensus method when they do this sort of review. And in that consensus method, they say, well, suppose I'm trying to compare against a particular suspect. That's where these red dots are coming from on top of the peaks. Those are Brian Shivers' genotypes, his allele pairs. And they ask, am I seeing the same sort of pattern, or the same alleles appearing at every locus? And what they see is there's nothing here, there's 2 matches there, there's 1 match there in the 3 different tests, A, B, and C. Each test, again, has 10 loci, so there's 30 tests altogether. And so while we have a lot of data, there's very little consensus between them. So the result of all this is that when a person goes to analyze it using a method like the consensus method, or some other way of looking at all this data, you can qualitatively see that there's an association between the passenger buckle and Colin Duffy. There's an association between the cell phone and Brian Shivers. And there's an association between the matchstick and Brian Shivers, but there's no actual match statistic. And why would you care? Well, if you go before a judge with DNA that doesn't have a match statistic, the judge can do one of 2 things. They can say, that's fine. I respect the expert's opinion that they think that these 2 things are matched and the person's in there. Or they can also say, there's no match statistic and that evidence is inadmissible. That's becoming more common these days. Ideally, you'd rather, whoever's proffering the evidence, you'd rather go in with a DNA match statistic that describes how much more specific this match is than some coincidence. So now I'm going to tell you what the computer does, because that was the point that Cellmark was at. They had wonderful evidence for the Northern Ireland police, but they didn't have any match statistics. And so the question was, how do you take the same data and conceptually the same DNA inference process of inferring genotypes and getting match scores, but instead produce a match statistic? So what's different about the computer is that the computer uses all of the data. People generally draw a line, and they say, well, in my lab, over that line, the allele is present, and under that line, the allele is absent. They don't make use of the quantitative data that says some peaks are taller than others. But the computer can use all those peak heights. It can use them whether they're above a line, below a line. It does computer modeling to determine the certainty of peaks. And it can use their heights as well. So the computer, the first major difference is that it makes more use of the data that's there. And this is how the computer thinks. The computer considers every possible genotype solution, or at least tens or hundreds of thousands of them. What it does is, if you see in blue over here, this is the allele pair from one individual, 6 and 9.3. That's in blue. And maybe there's another individual, represented in orange, and their allele pair is a 7-7. And then if you take a certain amount, quantitatively, of the blue allele pair, and add it together with the 7 allele pair, there's 2 copies here, then you'll get a pattern, and that pattern can be used to explain the data. The computer tries all sorts of patterns. It puts these boxes everywhere. It'll put them where there isn't data. It'll try 11, 12. It'll try everything. Most things don't explain the data very well. They have a very low probability. And some patterns and mixture combinations of those patterns explain the data better, and they end up with a higher likelihood. And so by trying out tens or hundreds of thousands of possibilities of what the different allele pairs can be, what the genotypes are, how much is present, the computer does a very thorough examination of the evidence in a way that we would expect a calculator or a computer to do that a person doesn't do. A person going through steps like this, there are some labs that have protocols that are similar, would look at far fewer variables and take a day to go through one cycle that the computer could repeat 100,000 times. So this is how the computer's thinking. It's trying all these different possibilities. And then this is what we actually computed at Cybergenetics for this matchstick. So when we received this data, as what often happens with trials, this was in November of 2010, we were told we had 1 week to get everything done before a court date, which was continued, of course. But in that 1 week, we had these 3 items of evidence and a lot of samples. We had all these different multiple samplings of those samples that I showed you. We just had a lot of data. And instead of running, as we might usually do, maybe 2 or 3 computer runs, we ran 185 computer runs over the week, trying out different combinations, as I'll show you, under different conditions. And this is what we did. We took the 3 samples from that Cellmark had produced, and then we had the computer say, okay, look at those 10 signals as one experiment. Try it under different computer conditions. And that was A, B, and C. Now, with a computer, when you're in court, you usually offer more than one piece of evidence and try to explain it with some story, one would hope, right? And so the same thing can be done with mathematical interpretation of DNA evidence. You can take the first set of data and the second set of data, and the computer can look at those 2 together and try to explain all 20 tests. It takes a lot more computer time, but it can try to do that. And if the data are consistent and compatible, then that analysis makes sense, and you can get a lot more information. So each of those, we paired A and B and C, and A and C, and each of those were 20 tests. We could also combine all 3 data tests together, right, from the first sample, A, B, and C, each had 10, and have the computer go through and look at all 30 tests simultaneously. These computer tests range from about 7 hours to about 7 days. Fortunately, we have a parallel computer at Cybergenetics with 60 parallel processors, so it can be working on 60 questions at the same time. Otherwise, we wouldn't be able to respond to non-existent court date deadlines in such a responsive way. Okay, so here's the data again. This is what it looks like at one of the 10 loci. This is STO1, and there are 3 experiments. We saw test 1 before, but there were 2 other tests, test 2, test 3. Notice they're different. There's an extra peak here at 7 that's in the first test, that's not in the others. 9.3 is taller in test 3 than 6, but it's flipped in other ones. These are different random patterns, going in and grabbing the marbles, seeing what's there. Sometimes there can be multiple individuals, or it can be that extra alleles drop in. A lot happens with low template DNA. But what the computer has to do now is to say, suppose it's trying out the hypothesis that one allele pair from one individual is a 6 and a 9.3, say in blue, and in orange, it's also a 6 and a 9.3. How well does that hypothesis explain the data? If the computer tries that with different amounts, it'll move those bars all over the place. And of the roughly 100 possible allele pairs that a human being could have at the locus STO1, when it's all done, spending a day trying all this out, or 2 or 3 days, what it's left with is only 3 possibilities out of 100 that had any probability left over. And that's the genotype. And because there's uncertainty, you don't get one allele pair with mixtures like this, unless you could get lucky, but usually you don't, whether it's human or computer. you get a probability distribution. The computer's probability distribution tends to be more concentrated or focused in regions where the data has more support, whereas human review tends to diffuse that out a little bit and not get as much information. Now, this is a completely objectively determined genotype. There's no suspect up here. It's not looking at a suspect. It has no idea who you're looking for. When it doesn't know what problem you're trying to solve, except what is the genotype of this evidence, in fact, of these 3 different samplings of the matchstick. When it's all done, that genotype can be compared against the suspect, 10 suspects, or an entire database of 1 million suspects. It doesn't make any difference. It's objectively inferred without regard to a particular question yet. Now, to form a DNA match statistic, we compare it with a person. So, what is a DNA match statistic? You may hear on TV or read about 1 in some number of people. Well, that can be problematic because if you have a match statistic of a quintillion, where are you going to find the other quintillion people? Right? Sometimes you scratch your head about some of the statements. That statement makes sense under certain circumstances, but when you have touch DNA and mixtures, that's not the best way to think of it. The best way to think of it is, what did I learn from the data? What did I believe before I saw the data, which was in the population with these 100 possibilities at this locus? And what do I believe after the data? What's the probability? Having seen the data, what changed? Now, until this point, we haven't said what the suspect's genotype is. We went from 100 possibilities spanning the court to maybe these 3, but now we're going to compare with what we believed before. In brown, I'm showing you the population probabilities. About 100 of these, 1 for each allele pair. And we want to ask or answer, how much more does the suspect match the evidence than a random person? What did we learn by looking at the evidence? Visually, what you can see is that before we started looking at the data, the probability of somebody in this room having that 9.3 allele pairs, their genotype, was 13%. After the computer spent days looking at the data in this one computer run, the probability is now 82%, but that's the genotype of one of the contributors to this evidence. So there's a gain in probability. Why are we looking here? Because that's the suspect's genotype. Of the 100 possible allele pairs at this locus O1, in red, 9.3 happens to be Brian Shivers' allele pair. So we could have looked at anyone's allele pair, but he's the suspect, so according to the math, we only look at that allele pair. And then we asked, what's the ratio of the blue probability, what we saw after the data, the evidence genotype, to brown? That is, to the population random genotype. What was the change in probability? If it's not the right person, it could go down. If it is the right person, it tends to go up. We don't know. In this case, it went up by a factor of 6. And that ratio is the probability of an evidence match, 82%, divided by the probability of a coincidental match, 13%. And at this one locus, you have a number of 6. Now we're looking at 10 loci. So when, there's our number 6. So when we multiply together what happened at all 10 loci, not just at THO1, the product of all those numbers, some of them are, all those ratios are bigger than 1, suggesting support for the hypothesis that Mr. Shivers is included. Some of those can be under 1. Evidence can go either way, but when you multiply them all together, what you end up with is the full match statistic, the joint match statistic. And in this case, the match statistic was about 1 million. And the way to read this in English as opposed to mathematics, which is a foreign language, is a match between the matchstick and Brian Shivers is 1 million times more probable than coincidence, which is plain enough English so that we can it doesn't have to be understandable to a jury, but it's fortunate that it can be, right? That's how much more probable it is that this is a real evidence match to Mr. Shivers as opposed to coincidence. So TrueAllele, over this week and a little bit afterwards, because there was some more data there was more data thrown at us, and things always happen at the last minute. If you have a trial date, there'll be more work to do, always. We ended up with match statistics in the, that we reported of about 6 trillion between the passenger buckle and Colin Duffy, suggesting that he was in the car at some point. We don't know when, of course. A match statistic of 6 billion between the mobile phone and Mr. Shivers, suggesting that he handled the interior of the phone. And a match statistic of 1 million between the matchstick in the road and Mr. Shivers, again suggesting that he had touched this matchstick at some point. Okay, so that was the end of the fun part. Now I'm going into the boring law court part. Sorry, we have to do this. That's why you paid your $10, for some of you. So what happened? Well, a year later, in November of 2011, I received a phone call at 9:30 a.m. on a Monday morning saying, could I be in court the next day in Belfast, Northern Ireland? And so this is what happened. So I was in the witness box for 3 days. They let me out for lunch, I think. The first day was a direct exam. It was an interesting exam because there was so much evidence in this case. The trial lasted 7 weeks. I think there are 8,500 documents filed. This is as Justin told you, they had a budget and they spent it. So they had £1 million worth of stuff to use. And the second day and the third day were all cross-examination, which I'll get to next. But in the direct exam, the admissibility was rolled in with the direct examination. And also, and usually, all of this was done before the actual laboratory findings were introduced. So I guess an advantage here is there was one judge, not a jury, so he could keep track of all of this stuff. This is the sort of information that we covered, and I'll touch on what we haven't talked about already. We presented the DNA biology, what mixtures are, how people interpret data, probabilistic genotypes. And why don't I start there with probabilistic genotypes. So in addition to introducing the evidence about probabilistic genotypes and what they were, their match statistics, and explaining all that, we also had to be doing a Fry hearing, or it's called a voir dire. They don't have a Fry test. Fry was American. And so how do you establish that probability and genetics are well established? Well, you can go back 200 years to Laplace, who laid out the principles of probability, which really haven't changed at all from anything that we're doing nowadays with our computers. Mendel, when he was playing with the pea, remember the pea plants and the experiments, the different characters back in elementary school, you learned about this. He was using probability, and his genotypes were all probabilistic genotypes. So flash forward to a few years ago, the FBI SWIGDAM committee, the Scientific Working Group on DNA Analysis Methods, in their new mixture guidelines of how you interpret evidence in 2010, has a paragraph 3.2.2, that explicitly says that if you're using probabilistic genotypes, presumably on a computer, then do a validation and use them as a good way of interpreting DNA evidence. So we've caught up with wherever we were 200 years ago in forensics. And other organizations like the Standards Institute, ANSI is the International Standards Institute, and NIST, the National Institute of Science and Technology, it's the new Bureau of Standards down in Maryland. They have interchange standards of forensic data that explicitly provide optionally for probabilistic genotypes. So there's nothing terribly new about probabilistic genotypes. They've been around for 150 years. What about TrueAllele Casework itself? We described what it is. It's very thorough. It uses all the data. It looks at things tens or hundreds of thousands of times, trying out all possibilities. So it's certainly more thorough than people on the same data. It's completely objective, doesn't know a suspect, and it's typically more informative scientific studies on laboratory data done in collaboration with a number of crime labs show that on average the match statistic is about 1 million times more informative. Can be 1 trillion times. Sometimes it disagrees with the lab and says there was no support in the data at all. So, but whatever it is, it's trying to get all the information that's present in the data, usually about 1 million times higher match statistic. And we also show that we, in the TrueAllele system, rely on generally accepted scientific methods, and we provided a bibliography of about 90 articles. That was a survey of, we could have had 10 times that easily, but a survey of the literature, scientific literature, showing that all these concepts are standard and have been around for decades, most of them anyway. We also discussed TrueAllele validation. There's a legal requirement for reliability, Rule 702 there. Now you've had your law, right? Rule 702 that talks about the need for evidence to be introduced. It has to be reliable, then you can have a Frye standard, a Daubert standard, and so on, of how it's reliable. But how do you establish reliability scientifically? For that, scientific studies are done, usually called validation studies in forensics, and those were done. I'll mention 2 later on. We show that there are about 15 ongoing studies in various phases of completion in laboratories around the world that are arriving at similar conclusions. There are independent scientific presentations by scientists from organizations like NIST that have their own TrueAllele system, and then do a validation study and go give talks on it around the world. We also showed, interestingly, that in the last few years, scientists have begun to rely on the TrueAllele method. NIST, the standards organization, creates tubes of DNA that have standards amounts of DNA in them, like a certain amount of 2 people, and they'll send them out to laboratories so they can determine how good their procedures are. When the standards organization makes their mixture standards, as they published last fall, of the 3 ways they establish what the DNA is and how much is there in the mixture, one of the 3 ways is TrueAllele. So if a standards organization is relying on a system to create a standard, that indicates somebody's relying on it someplace out there. It's also being used as a laboratory instrument. There are papers that people have submitted and talks that have been given where they're not trying to test TrueAllele, they're trying to make measurements of small amounts of mixed DNA in their laboratory and use it as a better microscope to understand what they're getting in their data. So laboratory scientists rely on it. And we also provided a citation index that showed that there are over 200 articles by other scientists over the last 10 or 20 years that rely on this line of research. So for the word reliable, there's a lot of relying going on over here of scientists on our method. We also showed that there was, and this turned out to be important in Judge Hart's decision, that there was regulatory approval granted by the Forensic Science Commission in New York State. There are about a half dozen states that have a commission on forensic science, and what these commissions do is they regulate what public laboratories can bring in as methods. And like other states that have these, New York State has a DNA subcommittee comprised of about a half dozen eminent scientists, usually throughout the U.S., sometimes international. And these scientists assess reports that are prepared by the laboratory, and we help with that. And over about a 2-year period, the New York State lab in Albany kept coming back with more studies. At one point, they delivered a CD with 600 megabytes of original data, case reports, comparisons, and so on, of just information supporting the validation study. Parts of this have already been published. And after looking at study after study, in May of last year, the DNA subcommittee approved and made a binding recommendation for approval the use of TrueAllele Casework for the Albany lab in New York State. This was ratified a month later by the Forensic Science Commission, and a public laboratory now had permission based on years of vetting and validation studies to use the technology. There's also legal precedent. In 2011 alone, there were over 75 TrueAllele case reports that were filed on DNA evidence in criminal cases. And while I don't give that much testimony, I did testify last year in state, federal, military, and foreign courts about the method. And then, I'm sure you... How many were here last year at the Foley presentation? So last year, Anthony Krastik and I gave a presentation about the Foley case, which was the trooper slaying the dentist, John Yelenick, out in Blairsville. We talked about the law, what happened, and so on. As you can imagine, with a life sentence, there's an appeal. And this came before the Superior Court. And there was a judgment at the end of last year, which 2 days ago has become a published precedential Pennsylvania decision. And that decision in this case affirms that TrueAllele is a reliable method that can be used throughout the state of Pennsylvania, which makes things more comfortable for defense attorneys trying to exculpate their innocent clients, as well as prosecutors trying to implicate ones who might not be. The opinion itself is fairly interesting in that, as Senior Deputy Attorney General Krastik had presented during the Fry hearing in Indiana County 3 years ago, he said, TrueAllele is not a novel method. It's just it's a cobbling together of existing methods that are well established, that have been published by others, and are just being done on a computer instead of being done by people. He also said, in the event that you don't find that it's novel, let's have a Fry hearing. But he argued both, that we don't need a Fry hearing because it's not novel, and if we do, let's have one. It was admitted into evidence. The trial continued. What the Superior Court seems to have ruled, I'm not a legal specialist, so this is all online. If you type in Foley 2012 Superior, you can see the ruling. Is that they agreed with Prosecutor Krastik that TrueAllele is not novel, and because there's nothing new about it, it's just existing science reduced to computer practice, that perhaps Tony was right and there was no need for a Fry hearing. That's, again, I'm just a layperson on this. So you should read it and decide for yourself, right? But there is now legal precedent in Allegheny County and the rest of Pennsylvania. TrueAllele is also a published method, and here are 3 of the papers. This is a validation study that we published 3 years ago that worked with laboratory synthesized samples where we knew the answer and could compare with human review, whereas this study, which was published in November in the Journal of Forensic Sciences, done collaboratively with New York State, this study was done on cases, and a comparison was made between human review and computer review. This is where we found that with, assuming, forget which, there were 2 different ways of interpreting evidence. One was 1,000,000 times more informative. Another one was 50,000 times more informative, depending whether it was 1 or 2 contributors. And then other people around the world have published articles about essentially the same math and the same computer methods in England, New Zealand, Denmark. So this is a published method. If you have a junior high schooler, right, and they want to do an interesting science project, they can read all about these methods and do a science project. It might need some support or some help, but in theory, it's possible. TrueAllele is also readily available. If anyone needs this and you don't have it, if you're an attorney and you want a reading on some DNA data, you can call us. We have that. If your crime lab has it, maybe they're doing it already because crime labs, including Allegheny County, buy things like TrueAllele computers. We use TrueAllele in the World Trade Center to re-examine 18,500 victim remains, as well as 2,700 missing people, and compare them to help in identifying victim remains. It's been used in the U.S. and elsewhere, and it's available to anyone, prosecution, defense, or the court, or even to both within the same trial, which has happened. And most every description about what we do, including talks like this, presentations, publications, transcripts, we post as quickly as possible on our website. It's public. There's no secret about what the system's doing or how it works. Okay. Day 2. Well, the cross-examination started off in a reasonably friendly way. It was more that the science was on trial. So this was a typical question from Mr. McDonald, who was defending Colin Duffy. He points to some law, some admissibility decision, and he states, we all understand the laws of physics apply from things, entire solar systems, down to a grain of sand. But once you get to the atomic level, all of a sudden the rules change, the laws of physics don't apply, and we have to start employing guesswork. And he asked me, what did I think of that? Is it the case that it may be the case that what we do with TrueAllele actually just doesn't apply to DNA when we get down to a smaller level? Actually, show of hands, who thinks I agreed? Okay, you're right. Okay, so at that point, I launched into a response. Now, you should know this was a 5-day trial with exactly 500 pages of transcript from my evidence. I don't know how many tens of thousands of pages there were from everybody else, but there was quite a bit going on. So I'm not going to give you the entire answer, but I began. No, I don't. Let me address your analogy. When you move from the world of Newton down to the world of Schrodinger at a subatomic level, you move from classical mechanics to quantum mechanics. The laws of probability begin to apply to physics and answers to physics problems. They become probability distributions. Anyway, I went on for a few more pages. It goes on for 2.5 pages with a history of quantum mechanics, classical mechanics, where it comes from, and then DNA, how we move to the molecular level from large samples to small. Remember the sampling with the big, large amounts? All that got covered. So leaving out the historical context in the interest of time, because you might want to have some questions. Imagine we've skipped about 7 slides. So I'll just read this. But when you move from a classical model, whether it is in physics or in DNA mixtures, it's necessary, as we, fortunately not you, observed historically in physics and see happening in DNA, that in order to get reliable, accurate, and reproducible results scientifically, you need to move to probability. And that's the gist of it. You don't want to throw out your data. You want to use it, but in order to use it, you have to use probability, because that's a more accurate and that's the correct description of reality, and it makes the best use of the data. And so I concluded by thanking Mr. McDonald, which I'm sure he was grateful for. I said, so I appreciate your analogy of moving from classical physics to quantum mechanics, because that's exactly what we see with DNA mixtures as you move to uncertain data based on multiple individuals as well as low template DNA. So they were less happy with me on the 3rd day, and so we moved from putting the reliability of TrueAllele on trial to the reliability of the expert. Maybe you've seen this. I'm told that by defense attorneys it usually doesn't work out that well with scientific experts, but this is pretty mild. This is sort of at the beginning, and this is Mr. Shivers' attorney, Mr. O'Connor. And so I'm answering his questions. No, that's correct. And I'm showing it in red to give you a sense of the stridency in his voice. So that's 33%, isn't it? Isn't it? And I start answering his question, and he continues, can you follow my question? The prosecutor, wakes up and looks at this, no. He needs to answer his question in his own way. And then he continues, you have reported on 33%. I'm not involved anymore. The judge says, Mr. O'Connor, please don't interrupt the witness, though he sounded more like Justin than like me. The defense argues, he's given an answer. The judge repeats, please do not interrupt the witness. Somewhat sarcastically, the defense turns to me, would you like a calculator? And at this point, Judge Hart says, don't ask any further questions until the witness is answered. And then it got really ugly. And we ended at 4 o'clock on that Friday. And then the judge let me leave. I came back by video link. 3 days wasn't enough. I guess they wanted more. And then there was, as you can imagine, a motion to exclude. And the defense prepared a 27-page statement, 99 paragraphs. The first paragraph essentially said, the technology is unreliable. And the next 3 sentences said, the expert's unreliable. And they gave a lot of footnotes, details, and so on as they went through all this. So some time went by, and then on December 1st, Judge Hart issued a 18-page ruling on TrueAllele, which Justin was at live, right? You were in the court. And this is online. It's on the website. If you type in the CLE, you'll find it on our site, and you can read the ruling. And the last paragraph reads, I'm satisfied that the stage has now been reached in the case of this system where it can be regarded as being reliable and accepted. And I am satisfied that Dr. Perlin has given his evidence in a credible and reliable fashion. So he sums up, addressing the first paragraph. In the light of these conclusions, I can see no basis on which I could properly exercise my discretion to exclude this evidence, and I therefore admit it in evidence. So that was a nice outcome for science. Some of us had the view, here's science on trial. Are we going to go back to the era of Galileo? Or have we advanced maybe to the era of Newton? Where are we in history? And it seems we didn't rewind quite back that far. So last month, as you heard from Justin, Brian Shivers was convicted of the soldiers' killings at the Massereene Barracks. And Colin Duffy was acquitted in both decisions that the judge had made. He said the DNA was definitely what the lab had done, what the computer had done. There was definitely DNA identification, but in only for one of the 2 defendants was there established some link to the crime itself. But perhaps more importantly, long term, is what also appeared in the UK on the same day, which was the realization that these same DNA techniques, in particular TrueAllele, and the use of computer interpretation of DNA evidence that were used in the Massereene conviction could pave the way for future trials. And the reason this is important in Northern Ireland is that with terrorist, or as Justin had indicated by analogy, organized crime attacks, witnesses are threatened, juries are threatened, and witnesses don't come forward. So their investigations rely very heavily on forensics. But it's not all that different in Allegheny County, where about 90% of the homicides are related to drug activity, and witnesses don't come forward. So if you're going to solve these crimes, then you need better forensic tools, laboratories, computers, robots, people, whatever, in order to solve crime here in our own county. Thank you. Thank you.